Lots of sunshine around Long Island for today. Beautiful weather over by Garden City. We'll have temperatures in the 70s. That's a look at your Long Island weather forecast. Now we're going to head over to our two top scientists, Taryn Wesley and Joseph Despino, with breaking news and a special report on insulin.
Thank you, Rich, and thank you everyone for joining us. I'm Joe Despino.
And I'm Taryn Wesley. Tonight, we have breaking news that will focus on the importance of the pancreas and its superstar hormone, insulin. Many people think of the pancreas as an organ that secretes pancreatic juice and aids in digestion. While this is true, what some may not know is that the pancreas can actually function as an endocrine gland that secretes one of the most important hormones found in the body, insulin. We will shed light on the insulin's structure and function. Then we will go into why insulin is such an important molecule and its crucial role in human health in terms of diabetes. First, we have to talk about the pancreas, which is located right here. This is the site of insulin's production. Let's take a look.
The pancreas is a soft, glandular organ that serves as both an exocrine and endocrine gland. The pancreas is located in the upper left part of the abdomen, directly behind the stomach and next to the small intestine. Normally, the pancreas is between 6 to 10 inches long and 2 inches wide. The pancreas is divided into 3 regions: the head, the body, and the tail. The endocrine portion of the pancreas is found within the body and the tail. One of the main functions of the pancreas is to act as an endocrine gland that controls the levels of sugar in the blood. The pancreas does this by secreting the hormones insulin and glucagon, which are produced on the beta and alpha cells of the islets of Langerhans, respectively.
Now that we have some background on where insulin is secreted from, we can begin to focus on why this hormone is so crucial for our survival. In order to understand its function, it's important to understand insulin's structure. Looking at the structure of insulin, we can tell it is a peptide hormone because it is made up of polypeptides. The structure shows 2 polypeptide chains, the A chain and the B chain. The A chain consists of 21 amino acid residues and the B chain consists of 30 amino acid residues. There are 2 disulfide bonds that link the 2 chains together. The first connecting the 7th residue on both chains, and the second which connects the 20th amino acid on the A chain with the 19th amino acid on the B chain. There is also an intrachain disulfide bond located on the A chain that links the 6th and 11th amino acids. As you can see here in this 3D model, insulin is synthesized from a proinsulin molecule that contain an extra peptide linkage. This linkage is cleaved in order for insulin to form. Here the A chain is shown in green, the B chain is shown in blue, and the disulfide linkage is in yellow. Insulin cannot function without its receptor, which is located on the surface of cells. Let's take a look at the structure of this receptor. Here is the insulin receptor. The receptor has an alpha subunit and a beta subunit that contains a tyrosine kinase domain. Insulin's receptor is composed of 2 alpha and 2 beta subunits that are held together covalently by disulfide bridges. The alpha unit provides the binding site for insulin and is located on the extracellular side of the membrane. The beta subunit is a transmembrane protein with domains at both the extracellular and cytosolic surfaces. Insulin binds to the alpha subunit, causes a conformational change in the receptor complex, and activates the autophosphorylation of the beta subunit. Once this is phosphorylated, the beta subunit is an active tyrosine kinase, which is able to phosphorylate intracellular proteins and can cause a number of cascade pathways in different cells. Insulin's receptor is very important because it allows insulin to come into contact with the cell from the outside and generate its effect on the inside of the cell. Let's take a look at how the insulin receptor works in the glucose transport pathway. This diagram shows how insulin ultimately decreases blood glucose levels by transporting glucose into the cell. When insulin binds to its receptor, the receptor becomes stimulated. The stimulation of the receptor activates the P13 kinase mediated signal transduction pathway. This pathway allows for GLUT4 glucose transporters to be translocated from the inside of the cell to the cell membrane. When they are in the cell membrane, the GLUT4 proteins are able to take up glucose into the cells and decrease the amount of glucose in the blood. Now that we understand how insulin's structure is related to its function, let's take a look at the overall role of insulin within the body.
When food is ingested, it travels along the digestive tract, where it is broken down into its component nutrients in order to be absorbed into the bloodstream. One such nutrient is glucose, a simple sugar. Glucose gets absorbed by the stomach and intestines and then enters the bloodstream. It travels through the circulation to all body cells. Once absorbed into the bloodstream, glucose circulates, causing the blood sugar level to rise. An increased level of blood sugar sends a signal to the pancreatic beta cells, which respond by secreting the hormone insulin into the circulation. Insulin is necessary for glucose to reach and be used by several important target tissues throughout the body. These include the liver, muscle, and adipose tissue. Insulin is necessary to keep blood glucose levels stable in the body. Glucose enters the cell through a process called facilitated diffusion.
We've seen how after a meal, when glucose levels in the blood are elevated, insulin is stimulated and released from the beta cells of the islets of Langerhans in the pancreas. We have also seen that from the pancreas, insulin can act on a number of different cell types. This includes the liver, muscle, and fat cells. Insulin is able to stimulate and inhibit many important steps in fat, carbohydrate, and protein metabolism in order to maintain glucose homeostasis within the body. Let's take a look and see how insulin is able to do this.
That insulin is produced by the beta cells of the pancreas and glucagon by the alpha cells. These hormones then enter the bloodstream and travel throughout the body. Although they act on just about every tissue in every organ, there are three particularly important sites. First is the liver. The liver, along with skeletal muscle, is responsible for energy storage in the form of glycogen. Thus, insulin's effect in the liver is to bring excess glucose from the blood into the liver cells, where it will be converted to glycogen, fatty acids, and triglycerides. Insulin also acts in the skeletal muscle, where it promotes entry of glucose and amino acids. Amino acids are then used in the synthesis of new proteins, while the glucose is used in glycolysis to produce ATP. Some glucose may also be converted into muscle glycogen, which is energy to be used later. Finally, insulin acts in adipose tissue, where it stimulates uptake of fatty acids, which get converted into triglycerides, which can then be used as long-term energy storage. As you can see, each of these processes are reversible, and wherever insulin stimulates a process, it generally inhibits its opposite. For example, in adipose, insulin stimulates lipogenesis and inhibits lipolysis.
Thank you, Darren. Insulin sounds like a really important hormone. Insulin and glucagon work antagonistically to maintain blood glucose levels, but if something is off and something is not maintained, it leads to a series of metabolic disorders, perhaps the most common being diabetes. Diabetes is broken up into three main types: gestational, juvenile diabetes, type 1, and adult onset diabetes, type 2. Tonight we're going to take a look at all three of them.
Regardless of the underlying cause of diabetes mellitus, blood glucose control is disrupted and glucose builds up in the blood to abnormally high levels, making the person hyperglycemic. Although there is excess glucose in the blood, the glucose cannot enter the cells and the cells are starved for energy. The liver recognizes the cell starvation and initiates gluconeogenesis, further increasing blood glucose levels. In hyperglycemia, glucose that should be used as energy within the body's cells is instead excreted through the kidneys in the urine. This is called glycosuria, a common sign of diabetes. Unable to utilize the blood's glucose, the insulin-deprived cells, starved for energy, begin to metabolize protein. This leads to the loss of intracellular potassium and phosphorus and to excessive liberation of amino acids. As osmotically active molecules of glucose remain at elevated levels in the blood, an osmotic effect between intracellular and extracellular fluid leads to severe fluid loss. Called osmotic diuresis, this dehydrating effect draws excessive fluid from the cells. As water and potassium pass out of the cells into the blood, this contributes to an electrolyte imbalance in which water loss exceeds the loss of glucose and electrolytes, contributing in turn to hyperosmolarity, or the abnormally elevated concentration of osmotically active particles of glucose in the cellular fluid. Hyperosmolarity encourages even more osmotic diuresis. A deadly cycle develops as water loss exceeds electrolyte and glucose loss, perpetuating dehydration, which in turn exacerbates the electrolytic imbalance, and so on. Excess fluid is eliminated by the kidneys. Osmotic diuresis often leads to frequent urination called polyuria. Polyuria, along with polydipsia, or excessive thirst, are two common symptoms of diabetes. Meanwhile, the liver converts the liberated amino acids into urea and still more glucose, contributing to further hyperglycemia and glycosuria, which leads to even greater electrolyte imbalance, even as the kidneys become increasingly unable to process and excrete the rising levels of glucose in the urine.
Gestational diabetes is a glucose intolerance that's first diagnosed during pregnancy, thus where the name comes from. Usually results from routine blood screening administered to all pregnant women. The beta cells of the pancreas are not functioning sufficiently enough to meet the insulin demands of the mother, which leads to hyperglycemia. The endocrine placenta is partially to blame since the hormones they secrete cause weight gain and other hormonal changes that lead to the body using insulin less efficiently. The excess glucose is transferred to the developing fetus. The fetus, of course, cannot use all of the extra glucose and therefore stores it as lipids. This results in an abnormally high birth weight known as macrosomia. Some of the negative effects of gestational diabetes may have on the fetus are an increased risk of dying shortly after birth, hypoglycemia, respiratory distress, and jaundice. The child will also likely develop type 2 diabetes and become overweight in adolescence and adulthood. Some of the potential health effects for the mother is the likelihood of having to have a cesarean section, preeclampsia, type 2 diabetes, and depression. Treatment and prevention involve healthy eating, physical activity, and if needed, insulin supplementation. We'll be right back after this quick commercial break.
I have one.
Not two.
One. I have one. I don't have two. One. I got one. My sister has type 1.
I have.
Type 1 diabetes.
I have. Type 1 diabetes. I have type 1 diabetes. I have. Type 1.
I didn't do anything to get it.
I've been checking my blood sugar 8 to 10 times a day for the past 10 years.
I had to learn to read labels so I can count all the carbs I eat. Diet and exercise aren't going to make me better. My body doesn't even make insulin anymore. This is my insulin pump. It keeps me alive. Now that I have it, I can play and ride my horse forever. I'm not afraid to take my insulin shot. This keeps me alive.
Type 1 diabetes is referred to as juvenile diabetes because most commonly diagnosed in children. Type 1 diabetes is also referred to as insulin-dependent diabetes. Type 1 diabetes is a T-cell mediated autoimmune disorder whereby cells of the immune system destroy insulin-producing pancreatic beta cells. The peak diagnosis is usually between 5 to 7 years of age or at the onset of puberty. Males and females are affected equally. The common diagnosis of diabetes is usually made based on the classic symptoms of polyuria, increased thirst, unexplained weight loss, and blurred vision. The cause of the disease is unknown, but suggests that genetically susceptible individuals that are exposed to environmental triggers can induce autoimmunity. The disease is influenced by genetic patterns, but has no simple pattern of inheritance. There is evidence that has linked the disease to defective MHC expression, and there is also evidence that viruses may play a role in triggering the disease. Complications of type 1 diabetes include neuropathy, nephropathy, heart disease, eye disorders, and skin conditions. A typical male with the disease will lose 11 years of life expectancy, while a female can expect to lose 13 years. Obviously, one would prefer to prevent the disease rather than treat it, but unfortunately there are no known means of prevention. Current treatment involves insulin supplementation, carb counting, and blood monitoring. The insulin injections and blood monitoring are extremely disruptive to a normal lifestyle and can also be quite uncomfortable, particularly in children. In January of this year, scientists announced a groundbreaking new study involving stem cells that could lead to a potential cure. Harvard researcher Doug Melton had been working on a cure since his son was diagnosed with the disease in childhood. He has been able to halt diabetes in mice for 6 months, and his team is now looking to replicate this in humans. The process his team developed involves manipulating stem cells to differentiate into human beta islet cells. The cells are then implanted into the subject and they immediately begin making insulin. In order to protect the cells from an immune response, they encapsulated them in a modified alginate derived from brown algae. This research shows significant promise for a possible cure in the near future. Type 2 diabetes is also referred to as adult onset diabetes or non-insulin dependent diabetes. This type of diabetes is usually diagnosed later in life, and it is a chronic disorder in which the body does not produce enough insulin, or it has developed an insulin resistance through the downregulation of receptors. Type 2 diabetes is usually diagnosed through screening in overweight people or people with high blood pressure. Blood tests would be the same as you would find in type 1 diabetes, along with an hemoglobin A1C level. Unlike type 1 diabetes, the cause of type 2 diabetes is known. It is caused by general bad health habits often associated with being overweight or obese, lack of exercise, increased consumption of carbohydrates, poor overall diet, age, race, and family history. The complications are the same as type 1, along with an increased risk of Alzheimer's disease. Another way this type of diabetes differs is in the treatment. Usually lifestyle changes such as healthy eating and calorie reduction, along with frequent exercise, can do the trick. Constant blood monitoring is still required, and in some cases possible insulin supplementation or other drugs are taken like DPP-4 inhibitors or GLP-1 receptor agonists. Type 2 diabetes can be prevented by eating a healthy, low-sugar diet, getting regular exercise, and having regular physical examinations. Well, that's our report on insulin. Thank you everyone for tuning in tonight. I'm Joe DeSpino.
And I'm Taryn Wesley.
Stay classy, San Diego.